All right, so let's get started. So today we're going to go over the practice test. So I hope some of you have had a chance to look at this test, practice test. Our real final, which is on Monday evening, 7 to 10, we'll look something like this. So as you can see, we will have like a total of 75 points for the final exam. So that's why I will put 25 problems in the final exam. And all of these problems will be the multiple choices. And I think for each problem, either I will give you like maybe five options or 2 options. If it is a true or false problem, so you will choose 1 from 2. If for other kind of like multiple choices, you probably have like about 5 options. There will be only 1 correct answer. So just choose 1. All right. So technically we have like 3 hours. So this is the routine for final exam. But based on my experience, yeah, probably you do not need that much time. So you can, most of the students probably will finish the exam by 2 hours. So which is good because we do not have that much, that many problems. So I'm gonna put the chi-square table there. So because the final exam will be a cumulative. Which means you're going to use some knowledges, principles learned from the beginning, of the class. For example, how do we use the chi-square statistic to test the second Mendelian law, right? So we want to see if 2 genes are independent and sorted, or in another way, we want to test if these 2 genes are linked or not. So this is equivalent. So we're gonna know, still we need to know how we use this table to find the p-value, and then we can, based on the p-value, we can make a conclusion for the hypothesis test. All right, so let's go over this example, 25 problems one by one. So the first question is is something about my my research. So I think at the last lecture on Tuesday, we so in the final slides, I think on the final slides I briefly described one project, research project in my lab. So we tried to develop a very highly efficient algorithm called the 3andMe to infer the chromosome length haplotypes using genomic data from single gametes. All right, so if you so you do not have to understand the entire thing, right? But basically, you know what the project is about. So what the project what the project is about, and then so I think, yeah, that's good enough for you to answer this question. But on the final, real final, definitely I will not ask the same question, right? So this is only for example. So because that research is doing the inference of the haplotype. Of course, it is related to the haplotype. So this question was asking which of the following statement is irrelevant to this project. So definitely D is not, right? Because this project is relevant to the haplotype. And we are using the gametes, so that's the haploid. And because of the crossing over and during the meiosis, you're going to generate different types of gametes. So that means like this projector will be related to, relevant to the meiosis and the crossover. Only this one, C, non-disjunction. And so this is non-standard event in meiosis, and which is not the focus of this algorithm. So that's why we need to select C, right? So this is just an example, general knowledge about science. The second one, all right. Which of the following is not true? Is not euploid? So this question is testing you if you understand the definition of euploid or aneuploid, right? Euploid means like questions. Let me see. Yes, exactly. I hope you can see my screen. So euploid means like so if we talk about the haploid genome, so which is 1N, right? So if you see in a cell, there's a 2N, 3N, 4N, and so on. If there's a multiple sets of the haploid chromosome, that is euploid. All right. So if you understand this, it's easy for you to identify the right answer for this question, right? B is not, because B is a trisomy 21. So this is 2N plus 1. It's not 2N or 3N, but this is not a euploid. So that's why we're going to select B. All right. Okay, number 3. So which of the following is not involved in the dihybrid cross of a standard diploid organism? So we're talking about standard, so no non-standard events, and this is a diploid organism. So dihybrid means like we have 2 genes, and for each of this gene we have hybrid. We have a hybrid genome, a genotype, right? So obviously we're going to select E because E is a hemizygosity. What is hemizygosity? Which means like there's only one copy of that genes, right? So this often occurs like there's one gene in X chromosome, but not on Y chromosome. That's why you only have one allele. So that's not the standard dihybrid cross for a standard diploid organism. So it's very easy, okay. All right, so the question number 4 through 7. So this is a little bit complicated. This one, I think this one is a little bit challenging. But still, if you if you review the second midterm and our lecture notes, so we can use what we have learned to tackle this problem. So it's complicated, but it's challenging, but it's doable. Sometimes, yeah, for this kind of challenging question, probably you need to spend a little bit more time on these questions in the final. But since we have plenty of time, so this should not be an issue for you. All right, so here, so we need to read the the problem very carefully to make sure we understand the question. All right, so we have like we have 5 yeast strains, right? So those are yeast, and so each one, so each of these 5 strains A, B, C, D, E carry different deletion, okay? But this is different. See, so this is different from what we have shown in the lecture. and what we have seen in the quiz. Okay, all right. So this deletion do not cover many genes. So in the lecture or in the quiz, we talk about a problem where the deletion covers more than one gene, right? But here, this deletion occurs within one gene. So it's in a very short region. It's within one gene. So there's one gene L, and the deletion actually occurs within this L gene, right? Okay, so now this is the data we have. So I don't want to go into the details about the problem, because this is the only thing we need to pay attention, because this is different from what we're familiar with, right? We have deletions. We do different experiments to get this data in the table. But the only thing that's the same thing, but the only difference in this problem is, okay, all the deletion region are within one gene. Okay, that's the key point. Okay, and all right, and there's like 7 point mutations. Okay, now so we have 5 deletions, then we have point mutations here. All right, we have 7 single base mutant lines. So if we okay, all right, or okay, so this simple single base mutation also occurs within the same gene, the L gene. So this is the problem. And let's see. Okay, so now in the table, this is the same. So we use a plus sign to indicate a wild type recombinance arose in this cross. So if we, so for example, if we cross the deletion A with the single base mutant 1, so we're gonna see 0. 0 means like this is the progeny from this cross cannot grow. Okay, 0 means minus, it cannot grow. However, if you see a plus sign, means what? Means like if we cross this deletion B with, for example, with the point mutant 4, we're gonna see this one is gonna grow on the minimum medium. Okay, so this is the question. All right, so now the first one, the number 4, the question number 4 is asking a very general basic concepts for this experiment. So which of the following terms best describe this nucing mutants? All right, so first, because look at this key point. So we are talking about one genes, right? So of course, this will be allelic, right? Because different mutant shows represent different alleles for the same genes, then the answer A is correct. And also the somatic is not because here we're talking about a yeast, so this is not somatic. Yeast is a single cell organism. All right. So now how about this two? So because all this either the deletion mutant or the single base mutant, they cannot grow by itself on the minimum medium, right? So what is the term to best describe this kind of like mutant strains? All right.
Auxotrophic?
Yes, very good. Okay. So this question is a little bit challenging. So probably, yeah, at the first look at this answers, probably you're gonna just choose this one. Actually, this one is also right, because we're talking about one genes, and each different mutant, either the deletion mutant or the mutation mutant, they represent different alleles. So I think, yeah, probably this one is quite challenging.
So we can expect that for this one, it's going to be one or the other when it comes to like what type of cell we're dealing with, and then what type of nutritional habits we're dealing with here.
Right. Yes, we're talking about different points about this kind of like mutants, right? So, so let me see. Yes, that's right. Okay. So I will not give you a lot of like questions like this, challenging questions like this in the final. But in case like for example, let's use this as an example. If you just choose this one, or if you just choose this one instead of the E, probably yeah. So we can give you partial credit. So we have 3 points for this question. So if you just choose this one or that one, so you can get half of the points. But if you choose the wrong one, so then you're going to lose all 3 points. I hope this is fair, right? And I think the problem 5 is no problem for you because we have learned this before, right? So I showed you how to solve this problem, the similar problems in class. So you just check the map here, and so look at the deletion region and compare this deletion regions with the data from this table, and you can easily find the right order of this 7 bases. So here we are not talking about 7 genes. In lecture, we talk about 7 genes, and you need to use this kind of experiment to find the right order of those 7 genes. But here we're talking about 7 single nucleotide bases. All right, so you need to find the right order. So I don't think I need to spend time on this, because everybody should be able to do this by yourself. Otherwise, there's a problem in the final. Okay, you will see a problem like this, and you need to know how to find the right order for this using map, deletion map, and the data in the table. All right, number 6. So this one is also very challenging, I think. So okay, so now if we cross, so we do a cross between deletion C, so deletion C actually is here, and the point mutant 1. All right, now let's look at here. So deletion C is like so the deletion C is involves the region of the 0.356, right? If you look at the right order here, 3, 5, 6 are in this region. So that's the deletion C. If we cross the deletion C, which means we do not have this, this region, and we cross this deletion C with another mutant with the point mutation 1. Okay. So if there's nothing happen, so this cross will generate, still generate auxotrophic progeny, right? If there's nothing happen. However, if something happened, what is the something I refer to? If something happened, so maybe there's a slight chance that progeny from this cross can become prototrophic. It can grow by itself on the minimum medium. So what is the event I refer to? So if there's something, what is the something I refer to? If during this cross, if something happened, so with a very small chance, some progeny from this cross may become prototrophic, and it can grow by itself on the minimum medium. Right. So actually, this is the answer for the number 7. So if there's a recombination between these 2 mutant strains, so for example, the deletion C has the deletion in this region, right? So but the mutant, the point mutant 1 has something wrong with this base. But if there's a crossover between 1 and 3, then if there's a crossover recombination between the precision 1 and 3, then there's a chance that some progeny will have intact gene, L gene. Then, so that progeny can grow by itself on the minimum medium. So I hope it is clear. So actually, this is the answer for the Number 7, so recombination between the alleles. Okay, the 2 alleles. One is the deletion allele, the other one is the point mutant 1 allele, right? And okay, so what is the chance of this phenomenon occurring? So because we know, okay, so we know the crossover is a rare situation, right? In the past, we learned if 2 genes are very far removed from each other, so this will increase the chance for the crossing over occurring between them. However, here we are talking about two loci within a gene. So we are talking about the probability of crossover within a gene. So this is a very small region. Yes, exactly. So this is the point. So the cross, very good. So this question, so if we cross the deletion C mutant with a point mutant 1, so for the majority of the progeny from this cross, they will be still auxotrophic, because most of them, if there's no crossover, right? Most of them either have the deletion of this region or they have a point mutation at this locus. However, there's a very small chance. The chance is very small, usually less than 1%. Why? Because this crossover occurs within one gene, right? So we are talking about the crossover between 1 and 3. So this is a very small region. It's within a gene. So the chance of having a crossover in this region is very low. So we're going to choose this one. So based on the common sense, all right. Actually, so I don't know if you can see, deletion C is not like this, because we need to, so the zeros, okay, so deletion C here, you see the 0 are separated. This is not right, because we do not know the right order for this 1, 2, 3, 4, 5, and 7, right? So once you get this right order, now you go back to see the deletion C, 3, 5, and 6, then this 3, so these 3 locus actually are next to each other. So this is the region, and the 1 is here. So the crossover between deletion C and the point mutant 1 is actually should be between 1 and 3. So only crossover occurring between 1 and 3 can generate the prototrophic progeny. Do you agree? Do you agree? So here, so in the table, those zeros are separated because we do not have the right order yet. But once you get the right order, so you can see that for the deletion C, these 3 zeros should be adjacent to each other. Does that make sense?
So right. You're saying that recombination between, say, genes 3 and 1, there would just be a really low chance of it happening.
Yes. So think about it. So this the entire region here actually is just within a gene, right? Gene L. So this is a very small region. So that's why it's very rare for us to see a crossover occurring in this region. So that's why.
Oh, so you're saying that there's a really small chance of it happening because of a recombination within a gene.
Yeah.
It has a really small chance of happening.
Right, yeah.
Then would any of these answers, recombination between, say, 7 and 4, 6 and 2, whatever, would any of them have like a big chance of happening at all, or would they all just?
No, the same, because I agree with you. So the distance between 1 and 3 is even smaller than the distance between 7 and 4, right? But still, we are talking about the region within a gene, so they are equivalently small. So that means like the chance to see any crossovers occurring within this gene,
incredibly small,
between 1 and 3 or 4 and 7, is very, very tiny.
Oh, okay. Yep. Okay. I completely understand now. Thank you.
Yeah, good. Yeah, this is So that's why I say this one is a challenging one because it's not it's different from the common problem we're dealing with, right? Usually we talk about the crossover between two genes, and these two genes may be a little bit far removed from each other, but here this is different situation. Okay. All right. Very good. Yes. So if they were further apart, yeah, of course. But if we still talk about within one gene, so this chance will be low. If like we're talking about two genes, so here we're not talking about the just point mutations. We're talking about like mutations within two genes, which are far removed from each other on the same chromosome. Then that chance. Then if we cross the two mutants like this, and the chance to see the prototrophic progeny appearing from this cross will be significantly increased. So depending on the distance, right? Okay. Okay, yeah, this one, yeah, I like this one, but this one is a little bit like challenging. But if you understand this one, so don't worry about the final. So the final problem will be less challenging than this one. All right. Yeah, this one is really like a little bit like, so it's still doable if you understand everything. Like, so I think, yeah, it's still doable, but it takes some like a brain power to get everything right. All right. So number 8 is not difficult because here we try to understand the two concepts, right? So one is the penetrance, the other one is the expressivity. So they have slightly different definitions. So here, if we talk about the degree or intensity with which a particular genotype is expressed, then this is the expressivity, right? The level or degree or intensity. So we're not talking about the probability that a specific genotype is expressed. The degree is related to the expressivity. All right.
What's the difference between expressivity and penetrance?
Penetrance is the percentage. So the chance that is so for example, yeah, I hope I can show that slide. So if two individuals carrying the same genotype, suppose they should show the color, the blue color. If both of them show the blue color, the penetrance should be 100%, right? If one show, the other one does not show, that's the 50% of penetrance. However, if you take a look at the blue color, if both individuals show blue, but one is the dark blue, the other one is like light blue. So in this case, when I ask you what is the penetrance, still the penetrance is 100%, but the expressivity is different because the blue color is more expressed than the light blue. Does that make sense?
So what you're saying is that penetrance, we're giving that an actual number, but expressivity, we're just saying, oh, it's a lot or it's not a lot.
Right. So when you talk about the expressivity, this is given that this phenotype has already been expressed, right? So if there's no blue color, we do not bother about the expressivity. If the blue has been shown expressed, then we can talk about how much blue this individual is expressed. Does that make sense?
Yes. Yes, it does make sense.
Okay, good. All right. So this is an easy one. So yeah, if you but you need to be clear. So I think, yeah, based on that explanation, so if you 100% understand the difference between these two, then you should be no problem when you see the similar problems in the final exam. All right. Okay. So this is example. So I could give you some definition problems, questions about other definitions. So that means like so over the weekend, please go over review the slides all the slides and pay attention to the important concepts that I mentioned at least twice in the lecture. So that might be the test point. Okay, just like this too. Yeah. All right, so this is for the inversion problem, right? So we have here, so we have a inversion. So this is parallel, right? Because the inversion is where is BCD, I think. No. So it's CDE, right? CDE. So this region. So this is this inversion region is does not involve the centromere. So this is a parallel, and so the question is like, so if you have a heterozygous genotype like this, if this individual is going to produce gametes, so which of the following, let me see.
Professor, before we go forward, the way that this question is formatted, that it says the answers say that there are 4 gametes.
Yeah.
So I'm guessing that there are going to be 4 different strands, you know, theoretically in this problem.
Yeah.
Would they all be would they be stacked in this order where it's lowercase, uppercase, lowercase, uppercase?
Sorry,
looks like,
say it again. Yeah, I didn't get it.
Because it looks like gamete 4 wouldn't be affected at all, because the only crossovers would be between 1 and 3 and then 2 and 3. And I just want to know if gamete 4.
Oh, okay. So here, so I say if all the gamete will be viable, so then. So I'm not sure if I get your question. So did you ask if this gamete can survive or not?
No. What I'm asking is since there are going to be 4 strands here, right?
Yes, 4 strands.
Can we assume that they're all going to be stacked the same way, so that I know what genes I'm crossing over here?
Yes.
So it's going to be lowercase, uppercase, lowercase, uppercase in this case.
Yes, because this represents a chromosome, right? So on this chromosome, for this 6 genes, you have like lowercase alleles, but for this chromosome, you have like the same six genes, but with different alleles, the uppercase, right?
Yeah, sure, but I'm asking what, like, if it's like if this lowercase one is strand 1, right? And this uppercase one is strand 2. Then can we assume that strand 3 and 4 are stacked in the same order? Where
it should be, yeah, it should be.
Oh, okay. Yeah.
I think I got your question. Yes, exactly. So what, to answer this question, you have to consider the order of these alleles in the parental type, because if the parents, the single parents, the genotype of this parent looks like this, so this means like this lowercase alleles are arranged in this order on one chromosome, but the uppercase alleles of these 6 genes are arranged in this order on the second chromosome. So if this is the situation, so you need to understand, so you need to be able to, you need to know how to draw the diacentric bridge structure, right? So I hope you still remember. So in the lecture, I showed you the structure with 2 centromere, and once you have that, so there's a break, random break on the bridge to produce 4 gametes, something like this. Okay. All right. So let's look at the question here. So following karyotype and the genotype chromosome 1, if during a single meiosis a crossover involves 1 and 3 occurs between an A and a B, and the second crossover, say between the D and the E. Okay, so and the following example, so which of the following example would be a possible genotype? So this is a little bit complicated than what we showed in the lecture, but still it's still okay. So let me stop showing this, and I'm gonna show this. So the key point is like this. So this is very similar to that question. As long as we understand what is happening here, so in that question, right? So we have like 6 genes, but on one chromosome we have lowercase, something like that. So this is a little bit complicated than this example, but still the same. So you need to know how to use this genotype information to derive this dicentric bridge structure. And then once you got this, so you don't worry about this the little fragment, because there's no centromere on this one, right? This will be lost. So only look at this dicentric bridge structure, and there will be like a random breakage on this bridge. Then we're going to generate 2 chromosomes, and finally we're going to generate the 4 gametes like this. So we will have like 1 gamete with the parental, 2 gametes with the parental type, and we will have like deletion product like this. So this is the key point. So once you can draw this kind of bridge structure, so then there will be no problem for you to answer that question. Okay. So if you, I don't want to draw the structure here, so but I will leave this up to you. So please try yourself and to see if there's any problem. So if there's still problem challenging for this one, so we can talk about this more tomorrow. So tomorrow afternoon, I'm going to host my office hours and the genetic clinics, so to help you if you have any questions. So this will be a fun question. So okay. Number 10. Let's look at the time. Oh, yeah, we have time. Don't worry. we have time to finish all the questions here. All right. So this is about the inversion. So the same, something like this. So this talk about like so on a standard genome in a corn, there are 2 genes. So one gene is this, the other gene is this. And if it is a standard situation, there are like 40 map units between these 2 genes on chromosome 1. However, if we cross the standard corn strain, corn line with another popcorn line, so this is a special popcorn line, and on chromosome 1, so there's a inverted region inversion between these 2 genes on chromosome 1, and an inversion, either the paracentric or the pericentric, if the region covers like 20 map units between these 2 genes. So if we cross these 2 corn lines, one is the standard line, the other one is the popcorn line involving the 20 map units inversion between these 2 genes. So if we use the data generated from this cross to derive the map distance between these 2 genes, what would be the measured distance? So this is the point. So we all know this, right? The initial the original distance is 40 map units, but there's a like 20 map units inversion. So this will become invisible. That means like if you use this cross as an example, as an experiment to get the data to calculate the distance will be 20 map units. 40 minus 20 equals 20 map units. All right, any questions for this one? Very good. Okay. Okay, next question 11 to 13. So we have a Drosophila, the fruit fly, and one gene is X-linked, and this gene will determine the eye color. So we have six alleles. So the wild type is always red. The wild type allele is red, but we have like 6 recessive alleles here, produce 6 different colors. All right. And if any of these 2 recessive alleles come together, so you're going to show the mixture of these 2 colors. So that's the question. All right. So based on this question, we have like several problems here. Okay, how many different hemizygous genotypes exist? Of course, we have 7 alleles, right? 1 wild type alleles and 6 recessive alleles. Then we're gonna have like, because we are talking about the hemizygous, which means like X, Y, right? Only X has this gene, but Y is missing. This gene is missing on Y chromosome. So that means like if we talk about the hemizygous, we will have 7 hemizygous genotypes. Okay, so how many different homozygous genotypes exist? Homozygous, the same, right? Since we have like 7 hemizygous, then if we double that, so we're gonna have like 7 homozygous genotypes as well. But this one is a little bit tricky, right? Now, if we talk about the heterozygous, which means we have 2 different alleles from this 7. So what would be how many phenotypes do we get? So first, we have a single wild type allele, which is dominant to all the other 6 recessive allele, right? If this wild type allele form a heterozygous genotype, then it will show a uniform wild type phenotypes, right? So that means like we have. So let's see. So maybe I can show you this. Second. Let me put a new slide here. All right. Okay, we have 7 alleles. We have a wild type allele. So let's use big A, and then we have 6 recessive alleles. Okay. So now, if we talk about the heterozygous, if the heterozygous has a wild type allele, so no matter which recessive allele appear here, so this will give you the wild type phenotype, right? So the same, right? Wild type. So this will give you just one phenotype, wild type. However, so if the heterozygous alleles are chosen from this 6 recessive allele, so each combination will produce a different phenotype, because as I mentioned, if heterozygous genotype from this recessive allele, we're gonna see the mixture of the 2 colors. What this means, like how many phenotypes we're gonna have? We're gonna have like this many. We choose 2, any 2 out of these 6 alleles, right? If you learn this in mathematics class, the combination, so this will give you this many phenotypes, which is 15. So this will give you 15 different phenotypes, and the 15 plus 1 equals what? 16. So I believe.
Wait, so yeah. So what you're saying is that every combination of every single one of those recessive alleles,
yep,
is going to give us a different phenotype.
Yes. So for example, so yeah, this is what we learned from the problem. So for the wild type, it's easy, right? It will always give you wild type. But if we choose like 2, 3,
sure,
4, 5, and 6. So this is something like the codominant. So for example, this one gives you like, let's say blue. This one gives you like a green. Maybe it's not. So I don't think so. It's scary. It will be scary if a fruit fly has a green eyes. But yeah, just use this as example. So if you have a heterozygous like this will gives you yellow. I think the mixture of blue and green gives it as a yellow, right?
It's like bluish green.
Oh, the purple. Oh, maybe no.
Or blue and yellow is green.
It's green. Oh, blue and yellow
is
yes.
green.
Okay.
Blue and green is just like
okay. Good. Thank you. Yeah. Okay. So yes. Okay. So if this allele will give us yellow, then if you put this will give us green. Okay. Now, if this one gives us like maybe not red, I don't think so we cannot use red here. So some other colors. So if you use 1 and A3, so this will produce some other colors here. So any two combination of this from this 6 recessive allele will give us a brand new phenotype. So based on this information, we can have like, this is the total phenotypes we can generate from heterozygous.
Sure, and we're not including like phenotypes where it's blue and blue and yellow and yellow.
Then that's not heterozygous, right? We're talking about heterozygous.
Oh,
yes.
You're completely right. Yeah, I was thinking of all the combinations, but yeah,
right. Yes.
Yeah.
Okay, so it's tricky. So sometimes, yeah, you have to read the problem very carefully to make it right.
Yeah, I definitely see what you're saying now. Okay, thank you.
Good. Sure. No problem. Oops. Okay, let's go back to the question. All right, so now it's obviously we're gonna select this, right? So this one.
Just one more question, Professor.
Yep.
So for the sake of this particular mathematical problem, what is there any sort of function on a calculator that we can use or equation that we can use to just super easily?
Yeah, that's a good question. So in the final, if I give you a question like this, so I think you can use your calculator. You cannot use your cell phone because cell phone is too smart. Smartphone is too smart.
Sure, but like what do you know maybe what function we should use or
the
what
combination function? So the combination function. So I don't know if you have learned this in math class. So usually the combination function in the calculator is C. One number, another number. So that means like you choose 2 out of 6 without considering the order. So yeah, that's the combination function you can use. Or because usually I would give you a very small number here. So you can just spell out all possible combinations by yourself. It won't be very difficult, I think.
Sure, I've got it. I know which function you use. Thank you.
Okay, good. All right. Yeah, I hope that helps. Yeah. Or if you do not, so during the test, if you do not remember what function you need to use, then I think, yeah, maybe you can just use scratch paper to just specify all possible combinations, and then yeah, then you can get the right answer.
No, I've got it now. I see the function. I plugged in the numbers and it works. Yep.
Okay, good, very good. Okay, nice. All right, so number 14. So this one is talk about the mutation. All right, so here we have like a neurospora. So now we talk about 2 genes, 2 different genes, A and B. They are totally dependent, and we have like a single point mutation in each of these genes, A and B, and the chance to get a reverse mutation, which means like it is a mutant gene, but the mutation can go back to the wild type. All right, but the chance is very low. So the chance is like 1 times the 10 to the -6. This is the chance, and this is the chance for both genes. So if you want to have a reverse mutation for gene A, that's the chance. The same for reverse the mutation for gene B. So now if we want to have the both mutation occurring, the chance will be what? The multiplication, right? Because they are independent from each other, A gene and B gene. So you just simply multiply these two numbers. That will give you the right answer. This one is not that difficult, right? Oh no. So the chance is like 1 to the 10 to the minus 12. But the question was asking. The question was asking. The question was asking like if you want to, so how many, so if the reversion rate for the point mutation is this for any genes, so how many spores, so how many progeny on average would the researcher need to screen to identify a single reverted with the genotype like both wild type for the wild type for both genes, right? So that means like you have to calculate 1 over the probability. So that gives you this many spores, the progenies. So is that clear?
So you want us
the same? So actually, I just asked the question in a different way. So if I ask you, like, what's the probability you're going to see a single progeny from this cross? What's the probability you're going to see a single progeny showing the wild type in both genes, right? So that's the 10 to the minus 12. But if I ask you in this way, so on average, how many progeny you're going to check to identify 1 single progeny carrying this wild type allele A and B. So you have to use 1 over the probability, which is this many progenies.
So what you're saying is that all we have to do is do 1 over 1 times 10 to the negative 6.
That's right. Yes.
On my calculator, it's saying 1 million. It's saying A basically.
1 million? You mean?
Yes.
So what formula did you use in your?
I use 1 over 1 times 10 to the negative 6.
Oh no, 1. So here we are talking about 2 genes. So you have to calculate so you have to use the formula like 1 over 10 to the minus 12 because we are talking about 2 genes here. You have to multiply this probability twice, right?
Oh, I see. I see what you're saying. Okay, thank you.
Sure. Okay. All right. Okay, now 15 and 16. So this one is easy because so based on this cross, so you're gonna figure out. So based on this these ratios here, you're gonna figure out the genotypes for different crosses, right? If you see this 9 to 3 to 4, so easy. So that's easy. That's the recessive epistatic, right? So we can immediately find the right answer. So it's no problem. And so if you got this, so you can understand the mechanism behind. All right, so we have 2 genes, and so 1 gene is standing upon the other genes. So we have learned to speak before. So yeah, just so let me know if you have questions. We can talk. So I don't want to spend time on this one. So but if you still have problem, we can talk about this tomorrow. And let's see. Okay, this one is very similar to the one. To the problem that I gave you in one of our quiz. So if you review our quizzes, so you're gonna see some question like this. All right. So which one is not true? The following statement. So let's look at the first one. Yeah, that one is definitely is not true. So, all right. So this is the parental type. If there's a single crossover occurring between S is here, and then the centromere. So if there's a crossover occurring here, so it will not change the relationship between S and R, right? So still give you the parental type, but this statement says a single crossover between S locus and the centromere will result in a tetratype, which is wrong. So of course we're gonna select this one. So the other statement should be correct. And okay, so now, so if this is the data, so if this is the data you see from this cross, and then if I ask you to calculate the distance between locus S and the centromere, so we're talking about the distance between S and the centromere, right? That's easy. So you need to find the M2 type from the results. So here M1 is S plus S plus SS, right? So that's S plus S plus SS. So that's the M1 type. The second column is the only M2 pattern. So the third column is still the M1 pattern. When we just look at the S locus, the fourth column is still M1 pattern. So only this one, the second column is the M2, and then we can easily use the formula to calculate the distance between S and the centromere, which is this, right? So this is easy.
Professor,
Okay,
I had a question on 18. The third, so the first column, that's M1. The last column is also M1. The second column is M2. What's the third column?
Yeah, this is so if we only look at S locus, so that's S plus S plus SS, still M1, right? So here we just look at S locus. But if we are talking, so if we are talking about R locus, so this one
that's M2.
Yeah, that's M2 for R.
Okay,
for
I was looking at both of them at the same time. I forgot that we're supposed to do them individually.
Right. We have to. Yeah, that's right. So yes. Okay. Any other questions for this one?
I'm not getting how we're getting 14 here, because it's out of 840, correct? And
so 840 is the total. So
yes,
that's the number.
So the equation in this occasion would be 140 over 2 divided by 840, correct?
Could you say that again? What is the formula?
The formula here would, in this case, it would be 0.5 times M2 over the total, correct?
That's right. Yeah, 0.5 multiply this number divided by so this number. Yeah, you have
I was
to,
looking at 140. My bad.
All right. So this number multiplied by 0.5, and then divided by this number. You're gonna get this.
Sure. Yeah.
Okay.
I was confused there. I thought you were pointing at 140 from.
Okay. Yeah. No problem. Okay. Good. Yes. Okay. So yeah. So you're gonna see some problem like this in the final. So make sure you understand the theorem behind, right? But yeah, I will not give you exactly the same problem, but yeah, something like this. Okay, so 19. All right, this is about the trisomic, right? Or the triploid. Okay, so we talk about this. If we cross, so this is a standard diploid, but this is a trisomic. So we have 3 alleles for this individual. So if we cross these two, so what is the likely the genotype and their correct proportion, the frequency of different genotypes? So it's very easy, because from here we know this individual, this parent, trisomic parent, can produce gametes. One third of the gametes carrying big A and small a, right? Another small a is contributed from this normal parent. So we're gonna have like 1/3 of the gamete from this parent showing the big A, small a, and another 1/3 of the gamete showing a small a from this parent. And for the little a gametes, so there will be like 1/6. The same. This parent will produce 1/6 the gamete of the big A. That's the gametes. The ratio, all right. So the ratio. So let me stop showing this, and let's go back to this. All right. So the ratio. So how can we get the ratio? Yes. All right, this is the ratio. So this is the ratio. Why is that? So the same question. So actually, this is the same with the question I showed you, right? So we have a parent, trisomic parent like this. So let's see what's the possible gametes can this parent produce. So if, for example, if the big A and so if the number 1 and the number 2 come together, so it will generate this genotype, right? And this will leave this one here. All right, this is 1/3 of the possibility to produce gametes like this too. All right, so if the 1 and the 3 come together, you're gonna get this and this. Still, there's 1/3 of the chance to get this. Actually, the genotype of this are identical. Genotype of this are identical, right? All right, now, so if 2 and 3 come together, you're gonna get a genotype like this, and leaving the A like this. So this is another 1/3. So the probability of getting this one is half of the 1/3, which is 1 of 6, right? The same one for this 1 of 6. 1 of 6, 1 of 6, 1 of 6, 1 of 6 of this. But you can actually combine these two because they are the same genotype. So that's why we got this. So 2/6 of genotype like this. So that's actually 1/3, right? So you can combine these two as well because the genotype of these two are the same. So that's why you got this. All right, and then you have 1/6 of this and 1/6 of this. So that's the frequency, where the frequency came from. So I hope this is clear. Okay, let's go back. Okay, so obviously, yeah, you can easily identify the correct answer by this. So if you understand how or what possible gametes can this parent produce and what are the frequency, then you can easily figure out this. Okay. Let's see. Yes, right. Because here, so we're talking about the cross, right? So this normal parent can always contribute a lowercase a. So the extra here, the extra here, the extra here, and extra here are from here, from this parent. Yes. Okay. All right. So number 20. From whom did you inherit your mitochondrial DNA? So that's obviously the maternal side, right? So you inherit the mitochondrial DNA from your mom and from your maternal grandmother. Yeah, so that's easy. And so this one is not so this one is a combined problem. It's not that straightforward, but still, like, so once you understand the combined points, it's not that hard to figure it out. Right. So, okay, the red-green color blind list is X-linked recessive traits. So this is X recessive, linked recessive. Mia has normal color vision, but her father is an affected individual. So because this is X-linked, so that means like there's only a copy on the X chromosome. Her father is XY, right? That means like her father, on her father's X chromosome, there's a recessive allele. Excuse me. Sorry. All right. Now, so Mia must inherit that X chromosome from her father. So she must have an X little b. Let's use the little b, lowercase b, to represent the recessive allele from her father. But Mia has a normal color vision. That means like she must inherit another big B linked on the X chromosome from her mom. So Mia must be a heterozygous, right? Okay, now Mia married with Pedro, and so Pedro is normal, which means like the genotype for Pedro will be X big B, X capital B, right? All right, now they have a daughter who has Turner syndrome. Turner syndrome, so I hope everyone understands this. Turner means like in this female, the daughter, she only has one X chromosome, just one X chromosome. And their daughter is a color blind girl. So which means like, what's the genotype for this single X chromosome? It must be X little b, right? X little b. And this X little b must come from where? From Mia. Because Pedro, her father, does not carry the capital B, right? So this answers the second question. So did their daughter inherit her X chromosome from Mia or Pedro? Of course, from Mia. All right. So why does this daughter only inherit X chromosome from her mom, Mia? Because she doesn't inherit any X chromosome from her father. That means like there's a non-disjunction occurs in the meiosis in Pedro. So I hope this is clear to you. So the logic, the reasoning like this can help you answer both questions. All right. Any questions about these two problems? Very good. All right, number 23. So this is the Robertsonian translocation.
Professor, I had a question.
Yeah, sure.
For Turner syndrome, can the mother ever have a non-disjunction, or does it always have to be the father?
No, so both parents could have the non-disjunction, but in terms of the specific situation here, so
yeah, it had to be Pedro.
Yes.
I was just wondering if there was any possibility that, you know, Mia could have had the non-disjunction, not necessarily in this scenario, but if a mother, like generally speaking, have a non-disjunction and
yes,
give a Turner syndrome.
Of course, okay. Yes, of course. I don't know. Probably it's not. This is not an equal chance for male and female, but yeah, I think yeah, both are likely, but probably with slightly different probability.
Okay, then. All right. Thank you.
Sure. All right, 23, the Robertsonian translocation. All right, so I hope everyone understands. So this is an important diagram. So I have this diagram in the lecture. So please make sure you understand this. All right, so this is one parent. The x-axis represents the carrier, right parent. This is a carrier. So so this parent actually has like 2n-1 chromosomes, right? And this is a normal parent, so that's that gametes from a normal parent. So if the kids from this marriage will have like 4 possibilities like this, 1, 2, 3, and 4. But this one is lethal. We have talked about this. So you will not see any born children carrying this lethal genotypes. So this is invisible. So which means like kids from this marriage only can show genotypes here, here, and here. So we have like only 3 possibilities. And the question is like, what is the chance that the child will be born to have the Down syndrome? So only this possibility, right? This is a carrier. So this is a normal phenotype, and this is totally normal. So then the answer will be 1/3. So only 1/3 of the progeny from this marriage will carry Down syndrome. All right.
So just to be clear, we're not going to include the lethal one at all.
No. So yeah, but in the final exam, if I give you a question like this, I will not tell you. Okay, you have to tell yourself this is lethal, and you are not going to see any progeny or kids be born carrying this genotype.
Sure.
Okay.
And
Yeah.
then what you're saying is any how do I put it? Any chromosomes that are stacked like this, where these are going to be lethal, specifically these.
Yes, because so for example, this one actually there's only one copy of the 21 chromosome, right? So that's lethal because in a human being, so we have to have at least 2 copies of the 21 to survive.
Sure.
But this one actually is equivalent to 3 copies, so that's the Down syndrome. So 3
Okay,
copies
and then
of
this
the
translocation carrier,
yeah.
it's equivalent to 2. It's just that the other one is attached to our 14th chromosome.
That's right. Yes.
And then this is our normal one over here.
Yeah.
Okay, gotcha. Thank you.
Sure. Okay, so it is very fun, right? Yeah, I like genetics because it's sometimes it's really fun, yeah, to tackle those problems. Okay, all right, 24 and then 25. All right, so this is like something like the chi-square test. Okay, so if we want to, if we do this like the hybrid cross, A gene and B gene both are hybrid, and if we got the data like this is the progeny. Definitely, we do not see the equal chance, right? So we have significantly more progeny carrying this genotype, which shows this will be the parental type, and this will be the recombinants. All right, then these 2 genes must be linked. So if this is true, so if we do the calculation, so you do not need to do the calculation because that takes time. So because based on this information, you can immediately know. So if you calculate the chi-square statistic, this number should be very large, something like this. So these are too small, close to 0. So this number will only occur when there's no linkage between these 2 genes, right? If there's a significant linkage between these 2 genes, you're going to see a big numbers like this, and the degree of freedom should be what? n-1, k-1. So we have 4 categories here. So the degree of freedom should be 4-1, which is 3. So without any calculation, you can find this right answer, right? And all right, so now, so based on this data, so what is the distance, genetic distance between these 2 genes? Of course, you're going to add up these 2 numbers because that's the recombinants divided by the total, which is 1,000, gives you 10 map unit. Okay, we're done. All right, so any questions?
How did you get the map units on 25?
Oh, 25. Oh, okay. So we have like this is the results from this test cross, right? A hybrid test cross. All right. So we have the first category is this. Definitely, this is a parental type. The second category is this parental type, and this will be the recombinant, and this will be the recombinant. So we have like this number and this number. If we add these 2 numbers, this gives you 100, right? And the 100 divided by the 1,000, this will give you the recombination frequency, which is the map distance between these 2 genes.
Okay, that makes sense. Thank you.
Sure. All right.
I had a question.
Yeah.
I'm sorry, I don't know if you already went over this, but is the entire exam multiple choice or just like half and half?
Entire. So okay. Entire exam. So I'm gonna give you the same numbers of the questions, the 25. Oh, nice. And because in the final, I remember for some questions I probably only give you like 2 options, either yes or no, true or false. So that would be easier. So most of the question were like this. So you will have like 5 choices. You're gonna select the correct one. So I have a question. I got a question about like number 15 and 16. Okay, you mean this one? 15 and 16. Yes. Okay. So based on this table, you're gonna see. So let's look at the last one. So this gives you the classical ratio, not the classical ratio, the ratio that is associated with the recessive epistatic interaction, right, between 2 genes. All right. Okay. So how can we get this this ratio. So let me show you. Maybe I can show you this. Give me a second. Find probably this one. Not this one. Sorry. Maybe this one. Nope. This one. Ah, yeah. Okay. So to answer the question for 15 and 16, so we have to understand this. Yeah, the final is actually the final is a cumulative, yes. But I some students also ask the same question. So it's hard to answer this question. So for the final we mainly focus on the third part. But for a lot of questions, if you want to answer the questions correct, you have to use the previous knowledge. So in terms of this, I think our final will be a cumulative. And also maybe you are going to see some very similar questions in the previous exam or a quiz. So that's why I highly recommend that if you have time, so please review your previous quizzes and exams as well. Okay, so the 9 to 3 to 4 ratio actually represent this recessive epistasis, right? In interaction. Yeah, it is cumulative. Yes, final it is cumulative. So I think it's safe to say that because you're gonna. So to answer some question, you have to apply some previous knowledge. So you cannot just forget about what we have learned before. So in terms of this, I think it's safe to say, okay, our final will be a cumulative one. Okay. All right. So I think this is I'm gonna spend the last 5 minutes to talk about this question, and then so I have another meeting at 11. So if you still have questions, we can talk about this tomorrow during my office hours and the genetic clinic. All right. So this ratio actually tell us, okay, this is a recessive epistasis, right? So this is the mechanism. So okay, so usually this will represent a pathway like this. So start from the colorless stuff. Let me see. Yes, that's right. Yes. So only focus on what we have learned, and especially for the stuff that I repeatedly emphasize. So those are the important things you need to pay attention. Okay, so this pathway actually start from some like the initial stuff, which is colorless, and then the gene 1 wild type will produce some intermediate product, which is the pink. And so if the second gene is also working, the wild type, you're going to see the end product, the blue, right? So that's why, so if you do the dihybrid cross, you're going to see, so dihybrid cross. So this 9 to 3 to 1 ratio must be the result from the dihybrid cross. All right, but so how can we link this 9 to 3 to 3 to 1 to this ratio? So this is the mechanism. All right, so if both genes are wild type, which is 9, then you're going to see the wild type phenotype, right? The blue pigment, the end product. because the pathway is okay. So the entire pathway is functioning. Oh, okay. Now, if we only have the first gene is wild type, but the second gene is mutant, which means we only see this intermediate product. We will never see the end product because of the deficiency of the second gene, right? The mutant. You're gonna see the pink thing. It's the pink flower. All right. On the other hand, if there's a mutant for the first gene, so the first gene is not functioning, and the pathway will be blocked at this step. So that means like you will never see either the pink intermediate product or the blue end product. So because of the mutant of the first gene, no matter what genotype the second gene will take, you always see the white phenotypes here. So that gives you, so that means like you're gonna group these two to get a full here. So let's go back to our question. So this is the same with that one. So if you want to get this, so the F1 should be dihybrid cross, right? Dihybrid cross means like the first parent should be this, something like this. This will not give you a hybrid cross, right? Look at the 3. This one will not give you the hybrid cross. This one will not, because so this parent is too, so the genotype is not clear, so this is not correct. And this will give you the D will give you the hybrid cross, and also the E will. But so by looking at the third one, you're gonna rule out B and C. You just look at A, D, E, because A, D, E are possible answers, and then you just look into this different. Then you can look at the second cross and then the first cross. You can rule out D and E, and only leaving A is the possible answer to this question. And once you got this, so it's easy so to figure out the pathway, just like the slides I just showed you. So you can easily figure out the right order of the pathway and where the genes is controlling each steps. I hope this is clear. All right. I think, yeah, so I have to go now. Sorry about this. So in case you have any other questions, yeah, please come to my office hours tomorrow. We're going to talk about that. Continue to talk about those questions. Okay. All right. Thank you very much. Oh, please, yeah, if you have time, please don't forget about doing the course evaluation. It is, I think the due date is tomorrow. So please let me know. Your general opinion about this class. So which is good. So I will keep for the future class, and which part I need improvement. So I will do better in the future classes. Thank you very much. Okay. Bye-bye. See you tomorrow. Bye. Thank you.